Kari, I'm going to start with you just to warn you, and I'm going to go this way.
Are you going to record us? What if we're wrong? He's going to.
Pressure's on. Pressure's high.
He's going to start. He's going to ask us. Okay. I'm too poor to print. Are you laughing at my quarantine?
Start with number 1. Okay, so here we have a frontal section of the heart, anterior view. All 4 chambers, all the stuff like that. And Akari, I'm going to start with you. Let's start by identifying this heart chamber right there. What's that heart chamber?
Right atrium.
That's the right atrium. Okay. And what do you know about the blood that is in the right atrium?
It's like oxygen to blood.
Oxygen poor. Okay.
Yeah, and more carbon dioxide.
And high in CO2. Okay. It's low in oxygen, high in CO2. Jordan, what's this vessel right here that's dumping blood into the right atrium?
The vena cava.
That's the vena cava. Which one is it?
Inferior.
This is the superior because it's coming from above.
Oh, down there is
Okay. And down here is the inferior vena cava. Ian, where is this? This is venous blood right here. Where is the superior vena cava collecting venous blood from?
Maybe the cephalic region?
It is the cephalic region. Pretty much the upper body. Okay, the upper body, all that venous blood drains into the superior vena cava, goes into the right atrium. Everything from the lower extremities and the trunk goes through the inferior vena cava and into the right atrium. Okay. Mackenzie, after blood goes from the right atrium here, where does it go next? What's this heart chamber right here? Into the what?
Right ventricle.
All right, so we got the right ventricle right here. And what's this structure that it has to pass through
Isn't that the tricuspid valve?
to get into the right ventricle? What is that? You're there.
Isn't that the tricuspid?
It's the tricuspid valve.
That's like the heartstrings.
Yep. So this is the tricuspid valve. There's another name for it. Anybody know the other name? I mean, it's more of a generic name, but I'll take either one on the lab exam. The right atrioventricular valve. Why is it called an atrioventricular valve? Separates the atria from the ventricles. Okay, Teddy, I'm going to go to you here and throw you guys some curveballs here. What are these little string-like structures that are attached to the valve? They are collagen fiber-like. Yep. Sierra?
What are they?
Yeah.
Particular?
What are the structures on the lab exam? If I pointed to these little guys right there, what would you tell me those are?
Or heartstrings.
They're your heartstrings. I'd maybe give you a quarter point on the lab exam for that.
Just a quarter.
Yeah.
Come on.
The chordate tendon. Those are your heartstrings. Pulling your heartstrings there.
Now, is that 2 words or 1 word for the chordate?
That's 2 words. There are these cords, tendon-like, chordate tendinate. And anybody, I guess, what are these structures right here that the chordate tendinae are attached to?
Papillary muscles?
Those are papillary muscles. That's cardiac muscle there, papillary muscle. Teddy, I'll give you another shot at this. There's blood in the right ventricle here. And where is that blood in the right ventricle? Where's that going? Generally. Where are we sending this blood in the right ventricle?
sending it up?
We're sending it up, and we're sending it to the pulmonary circuit. Okay, specifically, we're sending it to the lungs. Sierra, why is it that we want to send this blood to the lungs?
To swap out oxygen for carbon dioxide.
It's low in oxygen, so we want to get oxygen, and it's high in CO2, so we want to drop off CO2, so we're taking that to the lungs. Absolutely. Okay. Moen, what's the vessel system that this blood has to go through? First of all, what's this vessel right here? It's not the pulmonary arteries quite yet. Anybody?
Pulmonary trunk.
It's the pulmonary trunk. This is the pulmonary trunk, which makes these, what are these? Those are the pulmonary arteries. So you can think of it like a trunk of a tree, and then it has a couple branches coming off of it. Specifically, what pulmonary artery is this? Left pulmonary artery and right pulmonary artery. So this blood goes up to the lungs. We have gas exchange. And what's the blood like now? Caitlin, what's the blood like after it's gone to the lungs?
It's oxygen, right?
High in oxygen and
Low in CO2.
low in CO2. Okay? And I'm going to stick with you on this one. How does that blood go from the lungs back to the heart? What are the vessels that take it from the lungs back to the heart? There's two over here, and there's two over here.
Pulmonary vein?
Those are the pulmonary veins. Okay. These are the only veins in the body to have high levels of oxygen, low levels of CO2. These are the only arteries in the body, the pulmonary arteries, to have low levels of oxygen, high levels of CO2. Okay, Jennifer, where do the pulmonary veins dump their blood into? What chamber do they dump it into? It's this guy right here.
The left atrium.
What's that?
The left atrium.
The left atrium, exactly. Okay. And Marianne, where does blood go after it goes from the left atrium?
To the left ventricle.
Down here to the left ventricle. Okay, good. Molly, what's this structure right here that the blood has to pass through going from the left atrium to the left ventricle?
The bicuspid valve.
The bicuspid valve. That's one that I'll accept. There's also two other names for it.
Mitral and
The mitral valve,
atrioventricular.
left atrioventricular valve. Taylor, where does this blood go next after the left ventricle? Goes up to the aorta here. Specifically, this is the ascending aorta, aortic arc, and then everything after that is generically called the descending aorta, but for the lab, you're going to have to be more specific than that. This first part of the descending aorta is specifically the thoracic aorta, because it goes through the thoracic cavity. Then it passes through the diaphragm, and it becomes the abdominal aorta. And you have all these branches branching off. Ascending aorta, aortic arc, thoracic aorta, abdominal aorta.
Will there be arrows on the lab exam?
It's just a sticker.
We'll keep going with this because I want to call on everybody here.
After that one.
Audrey, what can you tell me? So here we have the atria, and here we have the ventricles. What's something that you notice that's different between the atria and the ventricles? Just as far as their structure, how they're built.
Bigger on the right side, and then there's the bicuspid and the tricuspid. I don't know why there's 3 on one side and only 2 on the other.
Well, let's talk about the atria relative to the ventricles here. Okay, here's the atria. Here's the wall of the atria. Here's the ventricles.
Ventricles are much bigger.
Ventricles are much thicker. And specifically, what part of the ventricles is thicker? What is all of this tissue right there that represents these big ventricular walls?
Cardiac muscle.
It's cardiac muscle. What do we call that? What do we call that layer? Anybody? That's the myocardial layer. Okay, that's the myocardium. I'll use that term, myocardium. Cardiac muscle.
Did you say myocardium?
Myocardium. Myo muscle. And Lilia, why is it that the myocardium in the ventricles is a whole lot bigger than that up here in the atria? Look, you can barely see, the little muscle layer in the atria.
Because it's trying to create more pressure to push the blood through the system.
It does create more pressure, and why is it though that the ventricles need to create more pressure than the atria?
Because the atria is creating so the atria contracts and it pushes the blood down and it makes a little bit of pressure in the ventricle. And the ventricles, when they contract, they create more pressure, which pushes the blood through the system down the pipes, both vessels.
Right. But what's the difference between the two systems there? What is it that...
Oh, there's more resistance in the vessels than there is going into the ventricle?
Yeah, that's basically it. So for blood to get from the atria into the ventricles, that's not very far to go, right? And as we'll learn in the next class, most of that blood that goes from the atria and the ventricles goes passively, just because of gravity. And then right at the end, the atria contract and they shoot that little bit of last blood down into the ventricles. But the ventricles, where do they have to send that blood? They have to send it through all these vessels of the body, and like Lily said, there's a lot of resistance there. So you need to create a lot of pressure to propel that blood through the system. Specifically, Lois, which of these has a large, if we just want to compare the ventricles now, which of these ventricles has a thicker myocardial layer, the right side or the left side?
Left side.
It'd be the left side. And
My brain, this looks backwards, but yes, left side.
and why is that the case? Why is it that the right ventricle wall is much thinner?
Because it doesn't have to exert as much force. It still has to exert force, but not as much as the apex.
It doesn't have to, and why is that the case? Danielle, why is that the case that the right side doesn't have to generate as much pressure as the left side?
Because going through the pulmonary trunk is a lot bigger area to go through, unlike going through the aorta.
The aorta is bigger than the pulmonary trunk. What about just proximity to the heart? Where are the lungs at? Lungs are just right here. So when the right side contracts, it's just got to send blood through the lungs here. The left side, where is that sending blood? Everywhere else through the body. So it's got to generate a lot more pressure to get that blood through the systemic circulation, is the key word there. What's this structure called right here? Anybody?
The septum.
Interventricular septum. Okay. We skipped a couple things. So for blood to get from the right ventricle up here to the pulmonary trunk, it has to go through that valve right there. What's that valve called? What's that, Sierra?
The pulmonary valve.
The pulmonary valve. Okay. And for blood to get from the left ventricle up to the aorta, it has to pass through a valve which is kind of right underneath there. What's that valve called? Anybody?
Aortic valve.
That's the aortic valve. Now, what do you guys notice that's different about the atrioventricular valves from these valves right here? We generically call these semilunar valves. But what's the difference between the AV valves and the pulmonary and the aortic valves? What's missing from those semilunar valves? The chordae tendineae are missing. Okay? So you guys are going to look at the pig hearts today, and they're going to be in this section. And you'll just open them up, and you're like, okay, you're trying to orient yourself and find yourself. These chordae tendineae really stand out, as do the papillary muscles. So it's very helpful because once you find those, you can be like, all right, these are separating the atria from the ventricles. And so it gives you an indication where the atria are above it, the ventricles are below it. And where are these papillary muscles and the chordae tendineae? Are those anchored up in the atria or down in the ventricles? They're down in the ventricles as well. So you guys will see all of that, and you'll know that this is the ventricular region. Now these valves work a little bit differently from each other. Blood goes into the right ventricle here. The right ventricle contracts, And what does that do to this valve here? It causes it to close, like so. And what role do these chordae tendineae and these papillary muscles have at that point?
They keep it from reversing back into the atrium.
I guess to prevent blood flow back.
They are preventing backflow. These valves are all one way. And so what happens is when the ventricles contract and the blood gets forced up through the pulmonary trunk there, this valve snaps shut. What happens is these papillary muscles contract a little bit and it pulls down on those chordae tendineae like so. Okay, so it's kind of anchoring these valves down and preventing them from flapping back into the atrium, which is called a prolapse. And we're going to do a case study where a guy has a heart attack, damages one of these papillary muscles in the left ventricle, and because of that the valve fails, and now he has a prolapse of the valve and blood's not going where it's supposed to go. Now these guys work a little bit differently. They're also one-way valves, but the ventricle contracts. It shoots the blood up through these, what you call these cusps, like so. And then what happens is when the trunk and aorta fill with blood and the pressure gets really high, that blood comes back down and it fills up those cusps and it causes them to close shut. And so their blood goes through them, they open up, then blood fills them up and they kind of slap back together. So they work a little bit differently. Another thing that I want to point out is that the events on the right side of the heart and the left side of the heart are happening simultaneously. It's not like blood goes, boom, 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 boom. It's not doing this one at a time. Okay, what's happening is blood is simultaneously moving from the atria into the ventricles simultaneously contract and shoot that blood out of their respective vessels. Let's watch a little video.
Yes.
Good.
Yes. Really need to see that.
Single cardiac cycle, starting at the right side of the heart. In reality, of course, the two sides of the heart work together.
Now it's going to go through step by step, and we don't need to do that. But what I want you to see here is that both the atria fill up together, both the ventricles fill up together, both the ventricles contract and shoots blood out. So each of those is one cardiac cycle, one heartbeat.
This animation illustrates a single cardiac cycle, starting at the right side of the heart. In reality, of course, the two sides of the heart work together at the same time, but it is easier to understand if we look at them separately, because each side of the heart has a different function. The cardiac cycle begins as deoxygenated blood flows from the body into the right atrium via the superior vena cava and inferior vena cava. In the next phase of the cycle, the right atrium contracts, pumping blood into the right ventricle through the tricuspid valve.
The left side's doing the same.
Once the right ventricle fills with blood, the tricuspid valve closes to prevent blood flowing back into the atrium. The right ventricle contracts, forcing the pulmonary valve to open as blood is pumped into the pulmonary artery. Blood then travels to the lungs and the pulmonary valve closes. At the same time, the right atrium relaxes, ready to accept the next intake of blood. In the lungs, the blood picks up oxygen and releases carbon dioxide. Oxygenated blood flows from the lungs into the left atrium via the pulmonary veins. The left atrium contracts to pump blood into the left ventricle through the mitral valve.
Mitral valve.
Once the left ventricle has filled with blood, the mitral valve closes. Next, the left ventricle contracts, forcing the aortic valve to open, allowing blood to be pumped into the aorta, and from there around the body. The aortic valve closes to prevent blood flowing back into the left ventricle, and the left ventricle relaxes, ready to accept the next intake of blood. This animation illustrates a single cardiac cycle, starting at the right side of the heart.
Boom, boom. side's doing the same.
Once the right ventricle fills with blood, the tricuspid valve closes to prevent blood flowing back into the atrium. The right ventricle contracts, forcing the pulmonary valve to open as blood is pumped into the pulmonary artery. Blood then travels to the lungs and the pulmonary valve closes. At the same time, the right atrium relaxes, ready to accept the next intake of blood. In the lungs, the blood picks up oxygen and releases carbon dioxide. Oxygenated blood flows from the lungs into the left atrium via the pulmonary veins. The left atrium contracts to pump blood into the left ventricle through the mitral valve.
Mitral valve.
Once the left ventricle has filled with blood, the mitral valve closes. Next, the left ventricle contracts, forcing the aortic valve to open, allowing blood to be pumped into the aorta, and from there around the body. The aortic valve closes to prevent blood flowing back into the left ventricle, and the left ventricle relaxes, ready to accept the next intake of blood. This animation illustrates a single cardiac cycle, starting at the right side of the heart.
Boom, boom. And these pig hearts, one, they're huge, but you guys are going to see all this stuff on there. You'll see the chambers, the valves, the aortic valve you'll see, the pulmonary valve is a little difficult, but you'll cut through the pulmonary trunk here, look down there, and you can see the little valve. All this stuff.
Are the pig hearts that we get, are they from like really large pigs?
They must be.
Because doesn't it say that our heart is like the size of a
Yeah, and in
pig's?
fact, they've done heart transplants using a pig heart. They're so similar.
But then when you were holding the pig heart, it took 2 hands for you to like
Yeah.
hold the whole thing.
Yeah.
It's
I don't know if they-
It must be like those big full-on sows.
Maybe so. Maybe so. Okay, so now we're going to get into some heart physiology, which you guys are all excited about. And there's a couple questions that I want to answer today. One, what causes the heart to contract? It's a good question, right? And the second is, what determines the rate at which it contracts, and also how forcefully it contracts? All good questions. And the answer to that initially is that in humans and other vertebrates, the heart contracts on its own. You can sever the nerves that go to the heart. You can sever the blood vessels that go to the heart, remove it from the body, and it'll continue to contract on its own. And the reason that's the case is because the heart has its own electrical system. All this is the electrical system of the heart, and specifically this is called the intrinsic conduction system. And what this does is it rhythmically stimulates the cardiac muscle and causes it to contract. Just like skeletal muscle, cardiac muscle first needs to be electrically stimulated before it can contract. So this intrinsic conduction system is going to strategically electrically excite parts of the heart, and that's going to cause the heart to contract. This is we're watching this video here, but this is a heart transplant, and they're kind of demoing this different method for transplanting hearts. You know, before, I guess, if they harvested a heart, they put it on ice and then they transport it. And now what they're doing, I guess, is they're hooking up a complete blood system to the heart. And but what I want you to take home here is that this heart is removed from the body and it's pumping on its own. It's not like the nervous system is causing it to pump or anything. It's this intrinsic conduction system within the heart that's causing it to contract.
Once removed from the donor's body, the heart is quickly connected to the device through the aorta.
There it goes. It's just contracting on its own, doing its own thing.
heart beating right in front of me. It continues to receive warm blood, as well as a proprietary formula of nutrients. Conceivably, how long could you keep a heart beating on this?
If you can renew it can go as long as you would like.
Tara Hale, the VP of Clinical Development at Transamedics, showed me how the device allows clinicians to monitor the heart's rhythm and other vital signs every step of the way. So this thing lets you know that everything is going okay?
Absolutely.
And what happens if something goes wrong?
You have detection here and alarms that are going on to tell you that you're out of range, and you can immediately control it. You can change pump flow, you can navigate into menus to change the settings to actually treat this heart. So you have a very comprehensive analysis of how this heart is behaving, and you have a chance to improve that behavior if needed and when needed.
So I have two questions for you. One, how fragile is the heart? And two, can I touch it?
One is not very fragile. Two, yes, you can touch it, but it's not the norm during clinical practice.
Okay, not the norm?
By all means, you can touch it. This is a training part today, so by all means, if you want to feel the ventricle pulsating,
Might be a pig heart.
right side and left side, and you can feel the vessel itself, see the pressure in the vessel.
It's warm.
It's warm, it's beating, it's functioning. It's just functioning as if it's in your body.
And it's not just hearts which can be transported on this system. There's a similar study currently underway with lungs, which has yielded some promising early results. So far, it's cut the complication rate by more than 60% and improved the survival rate by 30%.
Yeah, the heart is super strong, all that cardiac muscle. When I was in undergrad, took an animal science physiology class, and we worked with sheep and did some pretty cool stuff with them. But one of the experiments was terminal, meaning the sheep wasn't coming back at the end. And so they're all anesthetized or whatnot. And there was an abdominal incision, and the professor, punched his hand up through the diaphragm. And the reason he did that is because we were able to go in and stick our hand up the diaphragm and hold on to that heart, and to feel it beat. And it's just to kind of get a feel of how super strong it was. It is super strong. Yeah.
So the classic D horror movie where they rip the guy's heart out of his chest and it's still beating in the guy's hand, is that
Indiana
actually
Jones, Temple of Doom?
Yeah, exactly.
Yes. That's real. That's
Okay.
for real.
I always thought that was just Hollywood.
No. So like I mentioned before, it's this intrinsic conduction system that's the electrical system that's going to cause the heart to contract. It's going to establish the basic rhythm of the heart. So let's look at some of the components here. You may have heard of some of this stuff. What is this little button right there? That's the SA node, the sinoatrial node. You guys heard of the sinoatrial node before, the SA node? Well, the SA node is the pacemaker. This is the little bit of tissue that's going to establish the heart rate for the heart. And when the SA node is firing on its own and establishing heart rate, it usually drives heart rate at about 75 beats per minute. And that's called your normal sinus rhythm. When the SA node is firing and it's establishing the heart rate. Where's that SA node located? What chamber is that? That's up there in the right atrium. Then you have this structure called the AV node, the atrioventricular node. So the electrical activity is going to go from the SA node to the AV node. It's going to travel down this structure called the AV bundle. Okay. Then it's going to branch and travel down this structure right there. What is that structure? That's the interventricular septum. So the AV bundle branches into these bundle branches, travels down the interventricular septum, gets down toward the apex of the heart, and then that's where all these nerves spread out, and you can see it's kind of represented here, and they spread out all throughout the ventricular myocardium, the heart muscle in the ventricles. And that last bit of nervous system that extends into the ventricles are called the Purkinje fibers. I just noticed that. Subendocardial conducting network. We just call them the Purkinje fibers.
Is that what you're going to want to call it on the lab exam?
Yep, Purkinje fibers. That's not on the lab exam because we actually won't be able to see this stuff. It's all it's in the tissue. But you will need to know all of those structures. So again, SA node, AV bundle, bundle branches, Purkinje fibers.
How about the bundle of His?
Bundle of His is the same thing as the AV bundle. In the notes, do I call it the bundle of His?
Yeah, it's listed to label it.
Okay. Yeah, AV bundle is good. I'll take either, but. What page of the study notes is that on?
7.
So all of this electrical activity that goes on in the heart can be measured by an electrocardiograph. And this is an electrocardiograph right here. You hook up yourself to these different leads. And this is measuring the electrical activity of the heart. It produces a recording called an electrocardiogram. Electro referring to electricity, specifically electricity of the heart. And you can abbreviate that either ECG or EKG. This is the electrical activity for one heartbeat right there. There's one heartbeat, there's another heartbeat, then it goes on and on and on. And for each heartbeat, a typical ECG has 3 principal, what we call deflections. So this is just electrical activity here on the y-axis. It doesn't matter, why it goes up or down or all that kind of stuff. But what's more important is that you're able to put these little deflections and associate them with actually what the electrical activity of the heart is at the time. So specifically, we have 3 deflections. We have a P wave, we have a QRS complex, and we have a T wave. And these are all associated with different electrical events in during one heartbeat. All right, I may just go ahead and read through some of this here. So, electrical excitation of the heart begins with the sinoatrial node, up there in the right atrium. And the SA node is the electrical, what we call pacemaker, that's going to initiate heartbeat and sets the basic heart rate. When the SA node fires, a heartbeat is initiated. Okay. Specifically what happens is when the SA node fires, electrical signals from the SA node spread all throughout the atrial myocardium, the heart muscle that's in the atria. And when that happens, when the atria become excited from this SA node firing, you get this little P wave right there. So atrial excitation, or more appropriately, atrial depolarization. Depolarization means when something is electrically excited. Atrial depolarization is represented by this P wave right there. This is showing the SA node. It fires and all that electrical activity spreads all throughout the cardiac cells in the atrium. What happens after the atria are electrically excited? What happens after muscle gets electrically excited? It contracts. So this is atrial depolarization, which is followed shortly thereafter by what we call atrial systole. Systole means contraction, diastole means relaxation. So the atria first have to be electrically excited, depolarize, and then they're able to contract. Atrial depolarization followed by atrial systole. Something I want to point out, this is a cardiac muscle cell. Lots of these guys in there. What are those? Mitochondria, producing ATP because the heart's always contracting, right? But the point I wanted to make is that, this electrical activity is spreading pretty much simultaneously between all these cardiac muscle cells in the atria. This is one cardiac cell. That's another one right there. They took away the cell so you can see the borders here. But the reason they're able to do that is because these cells are connected by these protein pores that we call gap junctions. And these little gap junctions allow electricity to flow from one cell to the next. Anybody know what ion is going to be traveling through these cardiac muscle cells that represents electricity in the body? Sodium. Why is it important that all of these cells become electrically excited at pretty much the exact same time? So it contracts simultaneously. Exactly. We want the atria to behave as a single unit. How come the electrical signal doesn't travel down to the ventricles? I mean, first of all, let's notice that, right? The atria become excited, but if they're all connected by these gap junctions, how come the ventricles don't get excited? First of all, why do you not want the ventricles to become excited? What would happen if the ventricles became excited at the same time as the atria did? All four chambers would contract at once, and that's not the most efficient way to pump blood through the heart. You want the atria to contract first, so it sends all the blood down to the ventricles. Then, once that blood is there, then you want the ventricles to contract. So you want a delay between those two. I just want to point this out. The reason that's the case is because there's actually this insulating system in the heart that separates the atria from the ventricles. So the atria would be right there, the ventricles would be below it, and this structure here, it's a fibrous ring structure, is basically an electrical insulator. And it prevents this electrical signal from going from the atria to the ventricles. And so that allows them to behave as separate units. So how's that electrical signal going to get down to the ventricles? Well, that's where the AV node comes in, and the bundle of His comes in, the AV bundle. Okay. All right, so the SA node fires, the atria become excited, and then the atria contract. And what happens is that when these atria become excited, it also excites the AV node, the atrioventricular node. Now this thing is electrically excited. Where's that electricity going to go? It's going to travel from the atria into the ventricles through this AV bundle there. So the AV bundle is delivering that electrical signal from the atria through that insulating structure and delivering it down to the ventricles here. Does that make sense? Okay.
Is the electrical insulator is not depicted in this?
No, it's not. It'd be right there, and it'd be right there. And so this AV bundle delivers it through that signal and into the ventricles. So now it goes to the AV bundle, travels down the bundle branches, and an important point that I want to point out is that when the electricity is traveling down these bundle branches, it's not exciting the ventricles. It's still contained within that nerve-like structure. Okay? Yeah.
So if something goes wrong with that, is that what causes heart arrhythmia?
This conduction system, if something goes wrong with it, it can cause all sorts of different types of arrhythmia and whatnot. We'll look at some of those. So it travels down the bundle branches, then that electrical activity goes out through the Purkinje fibers. And this is an important point. It's these Purkinje fibers that are spreading out all throughout the papillary muscles, all throughout the ventricular myocardium, and that's when those electrical signals from the Purkinje fibers actually electrically stimulate the ventricles. So the SA node fires, atria are stimulated, that electrical signal travels down the AV node, bundle of His, bundle branches, into the Purkinje fibers, and once that sodium reaches the Purkinje fibers, then it shoots out and it electrically excites the ventricles. And that's what this is showing right here. That signal traveling down, and then once it hits the Purkinje fibers, all the muscle in the ventricles contracts. And when that happens, when the ventricles depolarize, when they become electrically excited, you get this huge electrical event called the QRS complex. So that represents ventricular depolarization, electrical excitation of the ventricles. And what happens right after it gets electrically excited? It contracts. You have ventricular systole. Yeah, and then something else I want to point out is that those Purkinje fibers extend into the papillary muscles. So the papillary muscles can contract along with the ventricles, and when they do, when they contract, they shorten and they pull down on these chordae tendineae, which helps the valve stay put so it doesn't prolapse. Okay, so we've accounted for the P wave. We've accounted for the QRS complex. The last thing that we have to account for is this T wave. And what the T wave represents is basically when the ventricles electrically reset themselves. It's called ventricular repolarization. They reset themselves, and what does that allow for? It allows them to be electrically excited again. So when they repolarize, when they reset themselves, that's the T wave.
How does that happen?
What's that?
How does the,
Repolarization.
how does that happen?
Just like the nervous system, these cells have a resting membrane potential. I don't know if you remember all that kind of stuff, but there's a high level of sodium outside the cell, high level of potassium in, and it creates this negative potential in the muscle cells. And then after it becomes electrically excited, basically it's the job of the sodium-potassium pumps to pump the stuff back to where it was and reestablish that membrane potential. Yeah, recalibrates it.
Potassium.
Sodium-potassium pump action. This is just kind of an overview for you guys. It, all this stuff, and it shows the atria contracting, shows the ventricles contracting. What do you notice about the time that the atria versus the ventricles contract? Which one of those takes longer to contract? Ventricles. Why is that the case? Because you really want to squeeze all of that blood out of the ventricles. A big, long contraction, squeeze it out so it shoots that blood out. So Ian, on your point here, the SA node is not the only pacemaker. Okay? The reason that it sets the pace is because it fires the quickest. It fires at 75 beats per minute, faster than any of this stuff. So that sets the pace. It's the pacemaker. But is it possible that your SA node gets damaged and no longer functions?
Yes.
Absolutely. And do you want to back up in place, or what if your SA node fails? I mean, the heart just stops? I mean, would you want that? No, I mean, you want to back up in place. Okay, you want something else that'll reestablish the rhythm of the heart. So the next fastest pacemaker, anybody want to take a guess? Is the AV node. And when the AV node takes over, it's not quite as fast, but it does generate a heartbeat at about 40 to 60 beats per minute, which is enough to maintain adequate circulation. You probably wouldn't want to go out exercising or anything like that, you know, but it is enough where you can survive like that. If the AV node fails, then the pacemaker becomes, you know, the AV bundle, these bundle branches and all that kind of stuff. But they're a lot slower. They only beat at about 30 to 40 beats per minute. And that's not adequate to maintain circulation. I want to throw something out to you guys. If your SA node doesn't fire, what does that take away from the cardiac cycle, from your heartbeat? If your SA node doesn't fire, what's not happening? You're not exciting the atria, so the atria are not contracting. Does that seem like a problem?
Yes.
It kind of does, because that blood needs to get from the atria to the ventricles. And we'll address this next class, but the reason that's okay is because, like I mentioned earlier, most of the blood travels from the atria to the ventricles passively, by gravity alone. And like 80% of it with each heartbeat dribbles down into the ventricles. Then the very last thing that happens is the atria go boom, they contract and they shoot that little bit of last bit of blood down into the ventricles. But if you don't get that remaining 20%, you can still survive. Yeah. So defects in this intrinsic conduction system can cause irregular heartbeats. We call those arrhythmias. Lots of different types of arrhythmias. Here's a normal sinus rhythm. So I have a little activity I want you guys to do. This is a normal sinus rhythm. And let's just say that this is 6 seconds right here. And this is time on the x-axis. That's six seconds. The first thing I want you to do is, based on that, I want you to calculate beats per minute. What's this individual's heart rate per minute? Then I'd like you guys to look down at this one here, and this is an arrhythmia. Tell me what's missing, tell me what's probably wrong, and tell me what the consequences are. Go for it.
So your SA is probably not working right. Or pacemaker. Not sure pacemakers. I'm pretty sure that they still do the atrium. Yeah, and then just multiply it by 10. So it's 8. So the pulse, there's 8 beats in the box, and it's over 6 seconds, so multiply by 10.
So that would actually be on 40. Yeah, if you count that, so each of these are it's like, no.
It's just it's weird. I'm not sure. I think it depends on which graph paper you're using.
I don't know why you have your pulse. I guess I thought it was 5 or 10.
Oh, yeah, right, I've never seen that.
I'm just totally seeing it.
I wish I could all along that way. It was real. So I gotta figure out how many kids in there, and we gotta figure out. Yeah, about the time.
Well, I I already figured that out.
Seven pulses over 40 a minute.
Yeah, he was dead.
Yeah, so I think that's all of those days.
I know, everybody's saying that's stressing me
No,
out.
this is the study. It's just kind of similar to that.
Yes, that's why I was looking for
That's too much counting. I would just turn my head if I tried to figure out how much each box is worth.
All right, let's do a little bit of math here, first of all. Up there in that normal sinus rhythm, how many heartbeats do you see? 8. 1, 2, 3, 4, 5, 6, 7, 8. So we have 8 beats over 6 seconds. This is math you guys should all have to be able to do, cross multiplication. So we want to know how many beats there is in 60 seconds. Okay, so we have, what does that give us? 480 equals 6x divided by 6. And what does that give us? What's 480 divided by? Did I do that right? 80.